We're going to have a look at photosynthesis today. So this is chapter 4.2 from your textbook. Our key knowledge, our dot point that we're looking at from the study design is the inputs, the outputs, and the locations of both of the light-dependent and the light-independent stages of photosynthesis in C3 plants. So these are just our everyday plants that we find in Victoria, basically. Many of our plants are C3 plants. About 80% of our plants actually are C3 plants. But notice this little bit in brackets. You do not need to know the details of the biochemical pathway mechanisms for these processes. So there's a lot involved in these, and if you do go on to university and look further at them, things like the Calvin cycle, you will study in a lot more detail, but you're not needing to know that detail at this level at this time. The role of photosynthesis. So this is where we actually transform the radiant energy from the sun. So we're capturing the sun's energy and we're able to then store that in the form of a sugar such as glucose. This is one of the key factors of photosynthesis. Without this, pretty much all life would not exist on our planet because the sun's energy, we can't actually utilize that. We cannot get food from the sun. But the plants have worked out a way of capturing that energy and turning that energy into a form of chemical energy that they can then store as the glucose. That means that we can then come along and utilize that glucose when we eat the plants or when animals eat the plants, and then thus we eat the animals. The chemical energy of the organic molecules is actually a dense form of energy that can be used as an energy source as well as being stored as either starch in plants, so our potatoes, our starchy products, or glycogen in animals. So we can often use the oils, fats to store that energy as well, and then utilize that when we have not got a food source at hand to then break down those energy stores in our body and keep us going. So it's really an amazing process that allows life to exist on Earth. You can see in this one here, we've got the equation for photosynthesis. So we can write that as a word equation. Carbon dioxide plus water gives us glucose and oxygen in the presence of light and the chlorophyll. It's really, really important to make sure that on your arrow you show light and chlorophyll. Otherwise you will lose marks on your assessments if you don't include these two. If you get them in the wrong location, so if you write them either side as a plus, then you will also be marked incorrectly. So it's really important that you note that light and chlorophyll are not used up in the reaction. They are almost similar to enzymes in that fact that they're not used. So it's important to make sure that you have those above and below the arrow, not either side of the arrow. You can see here that this equation here, so we've got two different equations here. We've got this one here, which shows you 6 carbon dioxides plus 12 water in the presence of light, and chlorophyll will produce our 1 glucose molecule plus 6 molecules of oxygen plus 6 molecules of water. Note that this equation down here is a simplified version of this above. Note it only has 6 water here, and it doesn't have any water at the end here. I recommend you know this one here. Knowing the word equation is fine, but this one here will help you an awful lot more when you start to look at the light dependent and the light independent stages, and you need to know what goes in and out of them. This one here will help you out a lot more than this one down here because it does leave the water off here. This is your typical diagram that you would see in, let's say, a Year 9 science class, where you've got energy from the sun being absorbed by the leaf. You've got carbon dioxide from the air being taken up by the leaf. The stomata in the leaf will open and allow that to naturally diffuse into the leaf. And then as a waste product, you have oxygen released into the air, and you also have glucose produced, which goes into the phloem. Remember that from Year 11 biology, there are two different passages or vessels in the plant. One is xylem, which transports water only, and the other is the phloem. Remember, the phloem can actually travel backwards and forwards through the plant. Water only goes one way. So water gets sucked up through the roots, and with capillary action is then transferred all the way up to the top of the plant and out of all of the leaves in the process called transpiration. So that water is released from the leaf, so out of the stomata again. And this is where that equation from the page before comes in handy, with the water being released at the end, because we are actually releasing water from this process as well. And that's quite often left off here, so just keep that in mind. Okay, as I've touched on, photosynthesis occurs in two different stages, and we'll go into these in a bit more detail in a moment. We have our light-dependent stage, and this can only proceed in the presence of light. So it must have sunlight to be able to capture that energy and convert that energy in the form of light. So that wavelength of light, they're converting into chemical energy, and that's being transferred via electrons into the coenzymes. Those coenzymes are then pushed through to the light-independent stage. Now, we used to call this the dark stage, but we don't do that anymore because it quite often confuses people to think that that happens in the dark. Well, it doesn't actually happen in the dark. It happens without light. So it doesn't need sunlight to occur, but it has to have the light-dependent stage occurring, because if we don't have that light-dependent stage, we're not feeding those coenzymes into the light-independent stage, and thus it won't function. So these two are very closely linked. They have to occur simultaneously, but one needs the light, one does not need the light. The light-independent stage is actually the one that forms the glucose molecules. So that's the part that's converting the carbon dioxide into our glucose molecule. So the light dependent captures the sun's light, the light independent creates the glucose. We'll go into more detail in these in a minute. Our trusty little chloroplast. Our chloroplasts are the organelles where photosynthesis occurs. The reason being they've got the green pigment chlorophyll embedded in the internal membrane of the chloroplast. They're present in really high numbers in the cytosol of mesophyll cell layers. Now, if you think about a plant cell, you've got, I think I've got an image of it. Oh, there's the chloroplast, and there's quite a few of them located within that cell. We've got about 40 to 200 chloroplasts per cell can be found in there. So depending on what the leaf structure is and what type of cell we're looking at, it will vary, literally vary between the cells. Now you can see here we've got our typical cross-section of a leaf. So we've got the top cuticle here, and often there's a waxy layer on the top there to protect the leaf. This is our upper epidermis of the leaf. Then we have our mesophyll. You can see that there's two different types of mesophyll cells. There's a Palisade mesophyll cell and there's a spongy mesophyll cell. The Palisade are packed nicely up the top here, but the spongy mesophyll have quite a bit of gaps between them so that air can then travel through. You can see down here we've got the stroma and we've got our guard cell either side of here, and the guard cell still has chloroplast in it as well. This is our lower epidermis, and again there's often a protective layer on here, and we allow air to pass in and out through our stomata. The structure of the chloroplast you're going to need to know reasonably well. In our chloroplast we have a few key points. The first one is that they have a double membrane structure. So they have an outer membrane, this one here, of a phospholipid bilayer, so same as the cell membrane, but it's got two phospholipid bilayers. It has an outer membrane and an inner membrane, and there is a space between the two membranes as well. So we have that outer membrane and we have an inner membrane, and this is really important when we start to look at where the chloroplast originated from. We have these stacks here called granum, or if you're talking, so granum will be plural, grana will be singular. So we have these stacks of little pancakes here. Now each one of the pancakes is known as a thylakoid. So this little stack and the top of, or any one of these little coins or discs, or however you want to refer to these, one of these is called a thylakoid. Now he's really, really important because this is where the light-dependent stage of photosynthesis is actually happening. One thing to note, when cells like this are drawn, they're often drawn with a green membrane, and the membrane is actually not green. If you watch Ed Rollo and you have a look at Andrew Douche on Ed Rollo, he will actually talk about the fact that this is cellulose, often found in the cell membrane, and in this case here, this is a phospholipid bilayer. Now, phospholipid bilayer doesn't have any chlorophyll in it, so there's no reason for it to be green. The green colour is actually from the thylakoid. So within the thylakoid, we have the chlorophyll, and this is where the green should be, not here. So just keep that in mind, but yeah, odd fact. Inside the chloroplast, we then have the stroma. So the stroma is this fluid area. It's like a solution that everything's just suspended in. So this is where the second stage of photosynthesis occurs. So this is where our light-independent stage occurs. So our light-dependent stage occurs in the thylakoid, and our light-independent occurs in the stroma. So basically the solution that all the chloroplasts and bits and pieces are sitting in. And this is where our Calvin cycle occurs. The stroma lamellae, so that's this bit here that interlinks. This is a tube that links the different thylakoids with different grana, and that we don't go into that, but it can transport various different bits and pieces between the granum, so between the stacks. We have also the intermembrane space. So this is the space between the two membranes. So this is this area here. And when we talk about some of the processes that occur, especially when we look at mitochondria, sometimes these spaces are quite important as an area that certain reactions can occur in because it's segregated from the others. We get into a little bit about the origins of chloroplasts. Now, this is not specifically in your study design, but it's good to know just in case. But it's not part of the study, or it's not on the study design. And I laugh at that. But it's good to know. So chloroplasts are believed to have once been free-living photosynthetic microbial cells. So they were actually on their own and photosynthesizing before they then became incorporated and engulfed by another microbial cell. So eons ago, these little microbes were basically eaten up by another microbial cell, kind of like this, and it comes along and goes and eats up the chloroplast. And all of a sudden, they formed an endosymbiotic relationship. There was mutual benefit for both of them. The chloroplast was protected from protected from the environment basically because he was sitting in a nice little cell that was doing its job, finding food, doing all bits and pieces, and so it was protected. But on the other hand, the cell, the microbial cell that had actually engulfed the chloroplast was now starting to receive food from the chloroplast. So the chloroplast was sitting there doing its little job, producing the food and probably overproducing the food, which meant that the cell that had engulfed it was benefiting from that. So all of a sudden we find, now we're looking here at the origin of the chloroplast, and we've got evidence of what we call the endosymbiotic theory. And we've got some points such as the fact that there's a double membrane envelope around this. It's similar to the outer and inner membranes represented by a cell wall of some bacteria. And it sort of shows us that we actually have this process where as it's been engulfed, another membrane has formed around it. And so we have that fact that we've got this first membrane of the actual chloroplast and then a second membrane, which could have been the cell that engulfed it, actually engulfing and the membrane forming around it. So it forms this double membrane in the cell. The second thing, they possess DNA. So they actually have a circular double-stranded DNA within the chloroplast, and it's not enclosed in a nuclear membrane. That DNA is able to divide along with the cell, and it's not by mitosis, it divides? It actually divides through the process of binary fission. So a chloroplast does not divide through mitosis, it divides through binary fission. And that is very similar to, of course, what occurs in bacteria. They have their own ribosomes. So they have their own ribosomes that are like those found in bacteria. They're smaller and they're present in, they're smaller than those present in the cytosol of the host cells. So they're different to the host cell's ribosomes and they're found within the chloroplast only. Because they have their own DNA and ribosomes, chloroplasts can actually produce their own messenger RNA and proteins. So they don't need the cell to do this for them. They actually can do this themselves. And lastly, their sizes fall within the size range of bacterial cells. So all of this evidence points to the fact that we believe that chloroplasts may have originated before being incorporated into a cell. All right, we're coming into the light dependent stage here, and I've got a little switch up the top here just to remind you that we have power on, light switch on. We are looking at the sunlight here. So this is the light dependent stage. It must have the sunlight. If you remember from our little diagram of the chloroplast, that process is occurring in the thylakoid. So it's actually occurring on the thylakoid membrane of the, or on and through the thylakoid membrane of the grana. And it's involved in the capture of the sunlight and transformation of its energy into chemical energy of loaded coenzymes. So remember, we've got NADPH, so remember the P is for photosynthesis, NADPH, and we've got ATP, and of course we've also got the two corresponding unloaded coenzymes, NADP plus and ADP. It occurs through a pathway that takes place within the grana. So each of those thylakoids is where we have this light-dependent stage occurring. The electrons are able to then absorb the light energy and become excited. I don't know if you ever remember doing the fireworks prac where you burnt different salts and you saw different colours on the Bunsen burner. Well, those electrons are actually able to absorb light rays as well. And as they absorb light rays, they become excited and they jump up a level in their atom. So the electron shell, they actually bounce up a level, and that enables them to transfer their energy onto an acceptor molecule. Now, some of the key events that are involved in this particular process. We have the capture of radiant energy of sunlight by the chlorophyll molecules, and this occurs, as we said, in those thylakoids, so the little tiny discs. The electrons are then able to absorb the energy in the chlorophyll to become very excited. They end up in a higher energy level, and at that point they're then able to transfer their energy by splitting water molecules to produce electrons, hydrogen ions, and oxygen. Those hydrogen ions, we also note that they can be called protons because they have that positive charge. The passage of the high energy electrons down a chain of electron acceptors occurs, and during this, our electrons actually release their energy in stages. As they release their energy, the electrons and the hydrogen ions are loaded into NADP plus to give us the loaded form of NADPH. So that's a proton that's been added to the end of that. And here is our diagram. So we've got, this is basically our chemistry behind it. We've got NADP plus. We've added a hydrogen ion, so a proton. We've also added 2 electrons to form NADPH. So already you can see this is loaded up with 2 electrons and a hydrogen ion. You don't need to remember this, by the way. This is just an explanation as to what's going on. So this one here you do not need to remember or memorize. This energy is used to pump the protons, so the protons are the hydrogen ions, from the stroma to the inside of the thylakoid. So this creates what we call a proton gradient. And so we have more protons one side than we have the other. That passive movement of the protons then back down its gradient, so it wants to equalize. If we pump them all one side, they're going to want to come back to equalize that gradient. And so as they come back, it produces kinetic energy that's used by ATP synthase, an enzyme, to produce ATP from our unloaded form of ADP and a phosphate molecule. Now, if you have a look at Ed Rollo, he shows you this section really nicely. So I would highly suggest you watch the Ed Rollo session after you've had a look at this PowerPoint to go through some of these details in how he actually forms and shows you what's happening here. That ATP synthase enzyme actually turns as those hydrogen ions are pumped through or passively move back through it. It actually turns, and it's like a little rotating sort of like a wheel at the top that actually allows our ADP and our phosphate ion to be joined together and form our ATP molecule. All right, this is where we start getting into the nitty-gritty bits that you need to know about. You will be asked what the inputs and the outputs of our light-dependent and our light-independent stage is. As you can see here, our inputs, we've got our solar energy from the sun. Remember, this is not used up. It is just here and formulating the reaction. So the wavelength of light is utilized in this. Notice it's shown sort of light, sunlight color. We've got our inputs. We've got water. We've got ADP, so unloaded ADP and a phosphate. We've got NADP plus unloaded. And with the energy from the sun, we are able to then split the water into oxygen. We pinch those hydrogens and they get attached to our NADPH. Our proton gradient then is able to turn the ADP into ATP. So you can see here we put in water, ADP and NADP plus. The hydrogen ions from here are taken and popped on the end of there to form our loaded NADPH. Our ADP and our phosphate ion are using that gradient of the hydrogen ions, so we're using kind of like that differential basically, to produce our ATP, and our water is split. So now we have our waste product of the oxygen. We don't need the oxygen. So that's a waste product, but we're using the hydrogen ions off that water. So they are our outputs for the light-dependent stage. Now, your textbook doesn't actually show you any numbers as to what's happening here. Just around the outside here, I've got the details of what's actually happening here. And here you go. I've added the numbers next to these to actually show you how many of each molecule are being utilized and produced. Now, in terms of having to know exactly the numbers, I don't know whether you are going to be asked them. I'm pretty sure all they're going to ask you is the inputs and the outputs. So you need to just be able to say water, ADP and phosphate. Don't forget the phosphate. And NADP plus are the inputs. You need solar energy, so sunlight. Don't forget the sun. People forget the sun. And we are producing oxygen gas, 18 ATP, and again, you probably don't need to know the exact numbers, and NADPH as your outputs. So you may wish to jot those numbers on. They're not in your textbook, so you may wish to jot those down, but they'll be on your slides, obviously. This is what comes from your textbook, the inputs and the outputs. So you've got inputs on the left-hand side and outputs on the right-hand side. You've also got an explanation as to the reactions there, but again, you do not need to know the chemistry, so you do not need to remember these reactions, but it's there to show you what's actually happening, to show you where the hydrogen ions, the electrons, etc. are actually going. Now notice it says down the bottom here that we've got water is split to create half an oxygen molecule. Well, obviously that's why we need 12 water molecules and we produce 6 oxygen molecules, half. We've got sunlight, the initial input of energy to chlorophyll. we've got NADP+, that's our unloaded coenzyme, and the water is split to grab the hydrogen and add that on the end. It also has 2 electrons for each NADP+, so we've got a hydrogen and 2 electrons. So it's carrying energy now. It's grabbed that energy from the sun by splitting the water molecule, and it's now loaded and got energy. ADP is also loaded, and it now has energy. And our water has been split, so our oxygen is now a byproduct. Just a little note down the bottom here, Pi represents that phosphate group, so PO4, and it's got a 3- charge. So remember how we talked about also DNA having a negative charge? That's that phosphate group that's actually producing that charge. All right, our light-dependent stage. At the end of the light-dependent stage of photosynthesis, our chloroplasts have a steady supply of high-energy loaded ATP molecules. They can unload and transfer their energy to drive the energy requiring an anabolic reactions of the second stage, the Kelvin cycle. And this is the light-independent stage. So this is where our products of the light-dependent stage now get fed into the light-independent stage. Remember, ATP does not last for long. So it's a molecule that's unstable and it won't last in the cell for a long amount of time. So that's not very good English. It won't last for very long within the cell. And so we need to use it reasonably quickly, which is why it is pushed from the light-dependent stage into the light-independent stage. So these have to happen side by side. We also have a high-energy loaded NADPH coenzyme, and again, unstable if it's left for too long, that can act as a donor of hydrogen ions and electrons in the energy requiring reactions of the Kelvin cycle. So we've got loaded with energy, 2 products coming out of here, ATP and NADPH. The oxygen, we don't want anymore, we don't need it. So the cell is actually releasing that into the spongy area. So the mesophyll cell is actually releasing that into the spongy area, and it is naturally diffusing out of the cell where it's in a high concentration within the leaf, and it's diffusing to an area of low concentration outside the leaf. So this is why plants produce oxygen gas, because they don't need it. It's a waste product for them. This little video is brilliant if you want to have a look at this. So I've got it in the PowerPoint presentation. He goes through exactly how the photosystems are working. So you can see in the diagram here we've got photosystem 1 and photosystem 2, so named because this was found first and then this was second, but this is actually the first part of the chain, and it shows you how the electrons are actually transferred through. And you've also got this ATP synthase pump, which is what I was talking about, the little cog on the end of it actually producing our ATP. So here's ADP here, and here's our phosphate that will tack on the end here. So this video is for you if you're looking for a little bit more detail and understanding of the electron transport chain, which occurs and produces that energy. So this is where the sunlight comes in, it's trapped by the photosystem, and this is how the 2 electrons here are transferred through the process, and we actually create that energy. So if you're interested in having a look at this, it's great, but it's not something that you have to know for the exam. It is just a little bit extra for you to understand how and why this process is occurring. If you are absolutely keen on doing biology later on, this is a good one to have a look at, because you're guaranteed to go through this at a later stage. And when we start looking at the electron transport chain in the mitochondria, this starts to make a little bit more sense if you know how this one's working. It's a similar process in the mitochondria. So up to you if you want to watch this video, but it is fantastic to go through. Okay. Moving on to our light independent stage. Note our little switch here is now in the off position. So we're looking at no light. We don't need light for this particular part. This is where we talk about the Kelvin cycle. So this is the stage of photosynthesis, and this is where we are actually converting carbon dioxide molecules into our glucose molecules. So this is what we call carbon fixation. We're fixing the carbon from the gas, carbon dioxide, into a product that the plant can actually use, so the glucose. We need a particular enzyme for this. So our main enzyme in our C3 plants is known as Rubisco. And the reason it's written quite strangely like that is because it's actually short for Ribulose 1,5-biphosphate carboxylase or oxygenase. Now the carboxylase or oxygenase comes into effect a little later on when we look at the variations of photosynthesis. But for now, what we're looking at is this particular enzyme. Now, you need to know about this enzyme, and it's specific in your study design that you need to know what Rubisco is and what it does. So this is why we start to focus on this one. As well as essential enzymes to catalyze each step of the cycle, other key requirements are the supply of NADPH, so we can donate those hydrogens and electrons that it's got. We need our ATP as our energy source, and we're going to be forming glucose as an output. So that's our product at the end of this particular process. Again, you must know your inputs and your outputs of the light independent stage. So our inputs are going to be our ATP, our NADPH, and our CO2. Note the ATP and the NADPH have come from the light dependent stage. All right, so this has come from the light dependent stage and it's coming into the light independent stage. The carbon dioxide has come from the gas that's allowed to come through the stomata into the plant. So, as you can see, our loaded coenzymes ATP and NADPH are passed from the light dependent stage. These are immediately recycled and reused by the light dependent stage when they're spat out the other side as unloaded. So you can see they're loaded when they come in here. So the inputs are loaded. Their energy is utilized in the light independent stage to form our glucose molecule from our carbon dioxide. And they're spat back out the other side, unloaded. So we now have ADP and phosphate. We have NADP+, and we have our beautiful little glucose molecule here. These two head back into the light dependent stage and are loaded up again to come back to the light independent stage. So you can see how this whole cycle must work together in order to make sure that our light dependent stage and our light independent stage are both able to function. So this is in C3 plants, so I'll specify that because we do have some adaptations in our C4 and our CAM plants. Again, with the numbers, just to show you what's happening, our 18 ATP that came from our light dependent stage, our 12 NADPH, and we've got 6 carbon dioxide molecules. We spit out the other side, 18 ADP molecules plus phosphate, 12 NADP plus, 1 glucose, but somebody's going to be wondering where the water came from. Remember how I talked about the water? We also spit out 6 water molecules, and this is not even in your book. So those 6 water molecules are released also as a byproduct at this stage. Now, because it's probably not essential that you add that in, you are really only required to recognize that ATP, NADPH and carbon dioxide go in, ADP, NADP+, and glucose come out. But I know you like to look at the finer details of these sorts of things, and so just recognize that that water is also coming out of this reaction. On Enrolo, if you have a look at 5A, so I would really like you to jump onto Enrolo, especially for this section, and have a look at what he's got here, because he's got some beautiful diagrams that I don't have time to animate to be able to show you what's actually happening through the process of photosynthesis, and he goes through this in detail. So please have a look at the Enrolo. It is really, really useful for this particular topic. Again, with our inputs and outputs for the light independent stage, we've got our NADPH, so our loaded enzyme is now being unloaded, and those hydrogen ions and electrons are now being used to form our glucose. We've got ATP now being the energy being used to form, again, the glucose, and we've got carbon dioxide coming in, which is our carbon that we're fixing into our carbon dioxide. Now, have a look at the other side here. This equation here, definitely not balanced, but it is showing you what's actually happening here. So we've got electrons and our hydrogen ions that are being combined to form our glucose molecule. And of course, we've got our carbon, our oxygen, and our hydrogen that's actually being utilized here to form this. So you can see there's double the amount of hydrogen than there are to the carbons and oxygens. And of course, we're releasing water as well. So this is where the water comes from in the extra hydrogen and oxygen that is here. Okay, our Kelvin cycle. There are several beautiful videos, and again, I'll link these on Infinita. One of them is Professor Dave goes through the light reactions in the Kelvin cycle on YouTube, and he's a very good one to have a look at. There's also Bozeman Science, who's fantastic to watch. So if you have a look at his photosynthesis video as well, and of course, as I've mentioned, there's the Ed Rollo that I definitely want you to watch. That's a must. The Kelvin cycle is our carbon fixation. So this is where we take our carbon dioxide and we turn that into our organic molecule. So we create our sugar from this particular process. Our carbon dioxide is picked up by an acceptor. So this acceptor is RUBP, Ribulose 1,5-biphosphate. And you can see where Rubisco gets its name from, because this is the enzyme that catalyzes this process. So our RUBP, ribulose biphosphate, is a 5-carbon molecule. So you can see here it's got 1, 2, 3, 4, 5 carbons in it. Over here, this little pink thing is a phosphate group and another phosphate group here. We've got our hydrogens as our little white ones and our oxygens as our red ones. And you can see carbon dioxide as well. Here's our carbon and our 2 oxygens. So what we're doing is we're using this and we're accepting that carbon dioxide to form our next molecule here. And you can see here we've got phase 1, carbon fixation, phase 2, reduction, and phase 3, regeneration of the ribulose. So we need to make sure that we've got this product back again so we can accept the carbon dioxide. So this whole process goes around and around and around, and it's all driven by this enzyme Rubisco. So he's a really important enzyme in the whole process. There we go. The reaction is catalyzed by the Rubisco enzyme, and it produces an unstable 6-carbon molecule that immediately breaks down to form 2 3-carbon molecules, these guys here. The first stage of the Kelvin cycle is our PGA. So this is Phosphoglyceric Acid. And there's another diagram here. This one comes from Ed Rollo, as you can see. And this one, you can see, actually has the numbers of how many are being formed each time here. So this can actually help you understand. So this is exactly the same process here. It's just got the Rubisco in the center. This is exactly the same process as what's on the previous slide, or what's underneath, the picture underneath. But this one's just a little bit clearer to see all those various components and the number of those components. Also note that he's put in here how many ADP and how many ATP and NADPH to NADP plus, and again another ATP to ADP over here. So this is where you get your 18 ATP/ADP and your 12 NADP plus. So this is where they're actually being interacted here. So I really like Andrew's model here and his explanation on Ed Rollo. So I really do encourage you to have a look at that. Okay, Rubisco is the world's busiest enzyme. And so it's key to this whole process of converting our carbon dioxide into our high energy organic product. So carbon dioxide is inorganic. We can't use it. We can't do anything with it. It's got low energy in it. But through photosynthesis, we're able to capture that energy, convert it into glucose, and be able to then utilize that glucose in metabolic pathways. So you can see here the central metabolic pathways, that's where our glucose is heading off to. The enzyme itself is actually quite slow, but because we have a very high concentration of Rubisco in our chloroplasts, this enables the Calvin cycle to just continue to go because there's a lot of this. It's super saturated with Rubisco. So even though it works slowly, we've got a lot of it to be able to cause this reaction to occur. Note this diagram only shows one cycle. It shows one reaction of this whole process. For every molecule of glucose formed, we actually have 3 turns of the circle. All right, so there's 3 reactions for every molecule of glucose formed. We then come into the reduction process. So the reduction process is this section over here. And this is where our PGA molecules are reduced to form another 3-carbon molecule, the G3P, so the glyceraldehyde phosphate, by accepting hydrogens from our loaded NADPH. So here's our NADPH, it accepts that and forms this G3P molecule here. Now, as I said earlier, you do not need to know these molecules. You don't need to know this process, you don't need the details of how this actually occurs. This is just information for you to understand how this process occurs, but you should not need to know the details. In other words, they're not going to ask you what this product is or how many carbons this product actually has or that sort of thing. They're not going to ask you key details about this. They can ask you details about how Rubisco works, and we'll get into something called photorespiration, where we look at what happens when Rubisco doesn't work as effectively as it should. So we'll get into that a little bit later. We need a lot of energy to actually drive this process here. So we've got our ATP that's being involved here to actually drive this through. And our G3P molecules are the output of the Calvin cycle, with 2 of these needed to form a 6-carbon glucose. We then have the acceptor stage. So this is the last one. So the RUBP acceptor that is used up in the first step is reformed in a reaction in which 5 of these molecules are reassembled to form the 3 RUBP molecules that we needed. So it regenerates itself. So as it comes up this side here, it regenerates and forms this RUBP molecule ready to go again. And so the whole process occurs again. This is an anabolic reaction because, as you can see, we're putting energy into this reaction. We're putting a lot of ATP into this reaction, so it's anabolic. Remember, catabolic releases energy, anabolic needs and requires energy. The regeneration of that RUBP is really crucial. If we do not generate the RUBP, we can no longer accept the carbon dioxide molecule. So if this little guy up the top here is not reformed, we can't use the Rubisco to accept the carbon dioxide and start the process again. So it's really important that we basically put it back to where it started from. Equally important for the operation of the Calvin cycle is the ability or the availability of the loaded coenzymes. So we need our NADPH and we need our ATP. Without either of which, nothing would occur. Okay, we need the energy input and we need the hydrogen ions and the electrons. After we unload them, the coenzymes are then recycled straight back to the light-dependent stage of photosynthesis. So they're then reloaded back to the original stage. All right, our light-dependent stage and our independent stages, as you've now gathered, are very tightly linked. Some of the outputs from each stage have to become the inputs from the next stage. And as I've pointed out, one cannot occur without the other. Loaded coenzymes that are outputs from the first stage are the inputs to the second stage. They are then unloaded by using that energy, and from the second stage are recycled back into the first stage to be reloaded. If the supply and the recycling of either coenzymes is interrupted, photosynthesis will stop. So it's a highly regulated, very crucial process. Often they will give you an image like this in the exam and ask you to label it. So you need to recognize that this dark spot here is our grana, full of all our little thylakoids. So this is a stack of lots of little thylakoids and it's known as the grana. Over here we have our space in between. So this is your stroma and this is the space that all of that is occurring in. So we love to use our 3D diagrams, but in the exam they'll often put a scanning electron micrograph picture in there and expect you to recognize and know what that is. So make sure you do understand this and you can label this. You can see all the lamellae in between here, the little linking bits. You can see the stroma, which is the space between. You can see the thylakoid stack here called the grana. So remember, when they're stacked together like this, that's your grana. So don't forget to be able to label this one as well. You can see a lovely little diagram down here which has the light dependent reaction and it looks like a pancake stack. So you remember that this happens in the thylakoid. It needs the sun's energy. It's producing ATP and NADPH. So these are the two loaded that go into the Calvin cycle. Calvin cycle churns around. It needs carbon dioxide and it spits out glucose. It also spits out unloaded ADP, and our phosphate, and also NADP+. These go back into our light dependent, which needs the water and the sunlight, and of course our oxygen is spat out as a byproduct. Up here we've got a beautiful little summary of what's going on, and this is what you do need to know for the exam. You need to know that we put in water, ADP, and of course because it's ADP it must have the phosphate, NADP+, and sunlight. If you remember that our light dependent stage is charging everything up, it's kind of like a solar battery. It's a solar panel, it's charging everything up. So everything you put in must be unloaded, it must be uncharged. As such, we're splitting our water molecule and we're loading up our two batteries basically. Here's our energy supplier and here's our hydrogen supplier with our electrons. So whatever we're putting into this, it's kind of like a solar battery, we're charging it all up. So here's our charge bits. Of course, to make the glucose, we need to use what's in the battery. So it's kind of like us turning on our torch or whatever. We are using up that energy. So here we have to put in the carbon dioxide because this is where we're getting our carbon to produce our carbon for our glucose here. So we need a carbon source of some sort, and that's where our carbon dioxide comes from. So if you remember that we need a carbon source, we've already got our battery here. So we've got our ATP and we've got our NADP+. We've got our charge here. And we can charge up our glucose molecule from this carbon dioxide. So we're producing our glucose molecule here, and of course our battery's now used up. So our ADP and PI and our NADP+. Notice these are in red, discharged, and these are in green, they're charged. So they've sort of color coded it for you as well. So you do need to know this table here. You must be able to identify the inputs and the outputs of both stages, and they say that very clearly. So you must know that table. And there you go. I don't know whether you need the quantities, and I highly doubt they're going to ask you exactly for the quantities, but if you remember that photosynthesis equation that we started with, then you're going to have the quantities nicely. Okay, you've also got your ADP and your NADP+, and if you go back through your the slides, you'll actually see you've got 18 and 12. So if you remember the 18 and 12, then you're pretty spot on the money as to what you need. But I don't think they're going to be testing you on the numbers of these. We do need to know in terms of the ATP that's produced for our mitochondria, but I don't believe they're going to test you on the numbers for these, but you must know the inputs and outputs of both stages. All right, ladies, I will leave you with one last slide, and this is your classwork or your homework. One of these things I definitely want you to do is to have a look at this Ed Rollo So the 5A, the process of photosynthesis in C3 plants, I definitely want you to have a look through that. I'm also going to load this extension up here on photosynthesis. This is a really good one to have a look at and have a read through. But again, it is extension, so it's not necessarily going to be something that's examined. But if you're looking at getting really high marks for your exams, then this is something that I would definitely encourage you to read. So if you're really wanting to do quite well, this is the sort of thing that you need to also understand. Have a go at exercise 4.2 and the biozone. You've got 43, 44 and 45 to have a look at those ones there. If you've got any questions, again, just email through any of those questions that you might have and I will answer those as I can. Have a lovely day. I'll see you soon.